PhD candidate in animal science. And she's going to be talking another food topic and what we're eating and the importance of better understanding kind of the food chain. So with that, I will pass the floor to you, Tiffany, and you're welcome to start.
Thank you, Andrew. Hello, everyone. I'm Tiffany. Have you ever seen bacteria? What are they look like in your life? What are they look like in the nature? Maybe you will see them on the plant with disease, or maybe you will see them on the rotten fruit. They doesn't look really pretty. However, if you come to my lab, you will see bacteria can be art. So sometimes they will look like storm, Australian map, sandstorm, firefly, universe, or deadly on. They're amazing, right? However, where are they come from? How can I get them? And more important, what can we use them for? So we need to create from this beautiful picture for a while and go into the problem our lab is focusing and research for. Everything starts with this beautiful animal, chicken. Poultry product is quite important in Canada. You can imagine, like, we can use the chicken meat for grilled chicken breast, and my favorite is the chicken wings. I hope everyone like that. Also, you can use their egg to make some great appetizer. Also, use egg to make your favorite protein as your dessert. Unfortunately, this high quality protein might also raise some risk. For example, our favorite chicken breast might brought us with some Campylobacter, and this cute egg can bring you with salmonella. For most of the time, you will just feel a little bit uncomfortable and maybe some diarrhea, but sometimes they can be really serious and bring you to the hospital. So, in order to save our health, government and poultry community try to eliminate the prevalence of this pathogen by improve the biosecurity level. Like they have vaccination program. They can also do some environmental control to improve the hygiene. Also, one of the most important treatment is medication. In the past 50 years, the poultry communities are highly relying on a very specific medication treatment, antibiotic. With the use of antibiotic, we can highly decrease the pathogen level in the poultry product. And also, people are not that easy to get sick. However, with the wide use, or say overuse of antibiotic in both clinic and livestock, we create a lot of bacteria with antibiotic resistant ability. And when one bacteria with multiple antibiotic resistant, we can call them superbug, bug, not hero. With anyone infected with this superbug, by far, not a lot of medication can treat them, or no medication can treat them, and we might have no choice but watching the patient die. According to a report, it is estimated that if we do not control the show up of superbug or the spreading of antibiotic resistance, there will be 10 million people died every year by the end of 2050. We need to make some change. Therefore, for both clinic and livestock, they try to say no to antibiotic. In Canada, poultry community try to eliminate or stop the use of antibiotics step by step since 2014. However, a lot of research indicated that we cannot totally stop the use of antibiotic if we want really safe food. we really need the alternatives of antibiotics. Hello, everyone. My name is Zhixuan Feng. I am a PhD student from animal science. My project is prospecting the chicken microbiome for anti-infective probiotics. Yes, our answer for the antibiotic alternative is the probiotics. Actually, by far, there are a lot of well-commercialized antibiotic alternatives for chicken. And probiotic, for something you and I would use every day, is one of the most successful commercialized products. However, in our case, we want to move one step forward, which is if we want to introduce a bacteria with a special weapon that can targetedly kill specific pathogen, which this weapon, it is Type VI Secretion System. This system can assemble an apparatus on the cell membrane, deliver the toxic effector into the target cell, and kill it. Therefore, in our hypothesis, if any bacteria with this weapon and is targeting our potential pathogen, we can treat it, we can make it as our probiotic candidate and save and protect people's health. Therefore, our research goal is finding bacteria from chicken feces that have the potential to be probiotics. In order to find the most powerful candidates, we try to make our experiment like first step, try to find as much as bacteria from chicken we can. And the second, we need to see if this bacteria have this special weapon or not. Then see if it can kill our potential pathogen or not. Finally, we need to know if it really is safe to use. So we are going to do the whole genome sequence and assess their safety. So first of all, we need to know, we need to have as much as bacteria we can. In order to do that, we collect this chicken feces sample from different provinces in Canada. Totally, we collect 6 chicken feces samples. And when they arrive to our lab, we will culture them directly. However, just like human, if bacteria, we want bacteria to grow more, we want them to be happy, provide them a happy life. So in this case, for example, but bacteria is a little bit different from human. So in this case, some bacteria do not want some oxygen. So we provide them with anaerobic chamber to make them free with oxygen. And sometimes they have a little bit stimulation. We provide them with the blood culture bottle, sheep blood, and rumen fluid. And sometimes they just need different kinds of the nutrition. So we provide them this colorful agar. Totally, we decide 76 different culture conditions for our chicken feces. And from only 6 chicken samples, we isolated over 3,000 bacteria. And also, even it's the long and tedious process. In between, we still find some surprise. This is bacteria art. See, we go back. That's where we find this art from a lot of culture. And when we isolated these cute bacteria and identify their species. So at first we will identify their protein. If their protein cannot provide the answer we want, we will extract their DNA and send them for the sequence. Okay, then we have bacteria and we know their species and we need to find our candidates. To identify the candidates, first we need to know if they have Type VI Secretion System or not. In this whole system, there is a component we really want to see is CLPV, one of the most essential components in all the bacteria, in all the Type VI Secretion System. And then when we find the bacteria with this system, we will send them for bacteria competition. We try to mix these our candidates and our target pathogen, culture them, co-culture them overnight, and then resuspend them and culture them in the selective agar. in this selective ACAR, you can see if it's the pathogen, it's almost show totally dark here. And if it combined with our candidates and the spots turn white, which means that it is possible for become our successful candidate. Therefore, finally, we will do the whole genome sequence to assess their safety. And in conclusion, from this beautiful bacteria, we are able to find some successful candidates. And that's all. Thank you so much. If you want to know more, visit our website and follow our Twitter and Instagram. Thank you.